The great majority of the ocean's living matter, by weight, consists of microbes. More than all the lobster, fish, porpoises, and whales combined. Think about what will happen when these krill die. Will they all be eaten? And what will happen to the eaters when they die? Marine scientists have known for a long time that when fish die, they sink to the bottom. And by the time they get to the bottom, they've decomposed into their essential chemical elements. So one thing the profusion of ocean microbes is doing is decomposing all the dead material floating down from above. In fact, that's how all the fish nutrients get released back into the water, so they can be recycled and used by other fish. If microbes exist in the oceans in massive numbers, and since the oceans cover 70% of the Earth, can there be any question that marine microbes play a vital role in the health of the planet and the health of humanity? But what exactly do they do? There are many clues, but at present scientists know very little. This man has dedicated his scientific life to filling the vacuum. Stephen Giovannoni, a marine microbiologist at Oregon State University, grew up around the ocean.
So there's a real sense when you look out across the sea that here's a mysterious thing that is very big, bigger than most of the other things in your life. Those mysteries are important for people. The ocean was important for me in that regard. The thrill of the hunt really begins when you get the idea that maybe you're seeing something that is very new.
A generation ago, scientists knew almost nothing of microbes in the ocean. Most doubted there was anything new to discover out there at all.
There were plenty of skeptics. There were people that thought that the kinds of bacteria that we already knew about were a perfectly good example of what lived in the oceans.
Even as a graduate student, Giovannoni was convinced that was short-sighted. And he was excited by a new discovery of life in the deep. Over 2 miles down, seen here in animation, scientists from Woods Hole, Massachusetts, found underwater volcanoes spewing superheated minerals from the Earth's core. When these minerals meet the ice-cold water, they crystallize, creating huge undersea structures. Spotlights pierce the blackness. This is real film. There is no sunlight. Yet life blooms in the darkness. How? Microbes. Instead of sunlight, hydrogen and sulfur from volcanic vents provide the energy for microbial life. The bacteria become food for some of the larger creatures and form the base of the food web. Shrimp, fish, and crabs thrive in this oasis of heat and microbes.
But seeing what could happen there, that there could be this unique ecosystem based on microbial life, stimulated me to think about the kinds of mysteries that might be right under our nose.
On a 1989 research voyage to the Sargasso Sea, in the southwestern part of the North Atlantic, Giovannone sniffed the first trace of the scent of success, though he barely knew it at the time. As beautiful as it is, the Sargasso might seem a poor candidate as a place to find new life. The Sargasso is very low in nutrients. But that's the point. It's not unlike most of the large central areas of the world's oceans. And since that's most of the planet, it's what Giovannone wants to know about. What's going on out there?
This is what most of the open ocean really is like, where nutrients are very low in abundance. The reason for this have to do with the motions of water. As you get near the edges of the continent, cold water, deep water comes up to the surface carrying nutrients. That doesn't happen here in the center of the Atlantic Ocean. Sometimes those nutrient-laden waters are brought up by a really big storm, but most of the time they're held below by the warm, lighter nature of the water above. So warm water is lighter. It sits like a blanket over the cold, deep water carrying the nutrients, keeps it down there. This means that the ocean surface around us is very, very clear. It's the color of really pure water.
The low nutrient environment presents a challenge. Finding and identifying microbes is that much harder. Like all scientists, Giovannone and his team are forever optimistic. Something new may be found that will help them make their mark.
We didn't fit. There we go. There are a lot of bacteria out there, but in comparison to the continental shelves close to the coasts of the United States, this water has far fewer algae in it, far fewer nutrients, and the organisms that live here have to recycle what they've got.
Giovannone has a hunch that in the low-nutrient Sargasso Sea, evolution might favor very small microbes. Theoretically, smaller means better in such an environment.
Organisms of the open ocean have special features that enable them to grow and compete. And my favorite hypothesis is that the smallest of the cells have a certain advantage over everything else because their surface area is high relative to the volume of the inside of the cell. And it's the surface of the cell that the cell uses to capture nutrients out of the surrounding water. Those cells are going to grow the fastest.
Giovannone's hunch was astute. In one of his sample jars, he had captured the first specimen ever of a previously unknown bacterium. As if living the reverse of every angler's favorite story, he was soon to qualify as the man who caught one of the smallest bacteria known to science. He and his team were about to make discoveries. Giovannone and his team have returned to Oregon State University to pan for microbial gold among the samples of water from the Sargasso Sea. But how do you know when you've found a new microbe anyway? Searching with a microscope is not realistic because so many different types look the same. Imagine this is Giovannone's mystery microbe, unrecognized in a sample of the Sargasso water. When he looks, he can't identify his quarry simply by physical appearance.
We had to find some other way of identifying them. What we did was to become gene hunters. In place of the organisms, we began to hunt their DNA.
Giovannone chemically broke open millions of different microbes. The messy results were billions of different DNA fragments, all mixed together.
What we're looking at here is approximately 50 billion cells worth of DNA. So in order to be visible to the human eye, you need to have a lot of DNA on there, a lot of cells.
DNA is a complex genetic recipe that consists of 2 strands held together by 4 chemicals called nucleotides, which are like puzzle pieces. The length and arrangement of these 4 chemicals determines the identity of every living thing on Earth.
We removed the DNA from the microorganisms in the seawater, and we took single genes. We took single copies of genes from all that DNA, and we studied those single genes. And we did it by reading the code on the gene.
Giovannoni retrieved pieces of DNA with just enough chemical code to analyze. He compared these with an up-to-date database of existing codes, the genetic codes of all known living things. One by one, the DNA codes matched existing organisms. But one code did not match up. It belonged to an unknown organism. They had discovered the existence of the mystery microbe. They named it SAR-11, Sargasso gene number 11. Now the real work would begin. He had to find the whole microbe. It wasn't long before the search took on greater significance. Giovannoni's research team found SAR-11 in water everywhere they looked. And they knew it was a bacterioplankton, which means it is involved in some way with the supply of nutrients to algae, the oxygen producers of the sea.
How is SAR-11 related to algae? You could say that algae are one half of the cycle, and SAR-11 is probably the other half of the cycle. What would happen if you took away one half of the cycle? The cycle would stop.
Following Giovannoni's lead, other scientists found the same gene in ocean samples from around the world. Evidence even turned up in freshwater lakes. Even more remarkable, in every sample of water there were more SAR-11 genes than genes from any other organism. Giovannoni realized that he was on the trail of possibly one of the most abundant life forms on the planet.
Right now, all we know about SAR-11 is one gene. And we know that gene is out there in the sea.
Stephanie Conan is a graduate student in Giovannoni's laboratory.
We feel that it is one of the most dominant organisms in the open ocean ecosystem. And being a dominant organism means it's the most numerous one. We feel that it plays an important role in the food web in the ocean, and we would like to find out what that role is.
But so far they hadn't managed to grow SAR-11. And for over a century, the only way to study microbes was to grow them.
This is one of the oldest methods in microbiology. It's called a spread plate. And really all it is dropping water from the environment on the surface of a sterile plate that contains food for bacteria, separating the bacteria so that they grow up by themselves and form colonies that we can see with our eyes.
One week later, so many bacteria have grown that they become visible. For years, marine microbiologists believed that the colonies which grew like this on spread plates revealed all the microbe species in the ocean. The reality was different. Over 99% of all microbes on Earth will not grow this way.
I know more about SAR-11's genetic makeup than I do about its appearances or its activity in the ocean surface. And making that transition from studying genes to studying the cells is one of the challenges of our work. The real challenge today is to try to grow SAR-11. Something about these cells makes them unable to grow in a scientific laboratory, at least today, using the kinds of techniques that scientists have. I told Stephanie this, and I said, what would be really a great discovery, but challenging and require creative thought, would be to try to isolate these organisms for the first time. Do you want to do that project? And she said, yes.
What I do in the laboratory is I try to grow these unknown organisms, unknown bacteria from seawater. We are going to be testing different food sources for this organism to determine what it eats. And if we can do that, we can start determining what its role is in the food web.
Our hypothesis is that SAR-11 eats organic carbon, but what we don't know is what kind of organic carbon does it eat? What is it like to eat? And that's one of the things Stephanie has to figure out in order to make it grow.
If Stephanie's diet for SAR-11 does get it to eat, grow, and reproduce, they will be able to find out much more about what the microbe actually does. Unlike many microbes, SAR-11 thrives with very little nutrients, so Stephanie is tempting it with very small amounts of food, 1,000 times less than normal. But it's a stubborn microbe.
We think that the reason it's so difficult to grow in the laboratory is that possibly SAR-11 is a small cell built to live at the very low nutrient concentrations that are found in the open ocean sites that we study, and that it's precisely adapted to do one thing, and if we change anything, then it doesn't divide in the lab. So the trick for us will be to somehow or another reproduce exactly what's happening out in the ocean in order to make it grow.
Brian Lanoyle is looking for an alternative way to snag a whole SAR-11 microbe. His approach is once again to use SAR-11's DNA as the tool.
You're going to stain for 30 minutes?
For the plan to work, he must first succeed in getting a larger DNA fragment.
Oh, yeah, it's a really nice unit, too.
In the past, we studied small pieces, but we could never study the DNA of an organism as one big piece. But this device gives us a way of working with very large pieces of DNA. And if it works, then Brian's going to use those big pieces of cloned DNA as a way of making SAR-11 light up under the microscope. He's got everything worked out, but the one thing we're missing is these great big chunks of SAR-11 DNA. We've been unlucky, and that's how it is sometimes in science. We keep missing it. We get other pieces of DNA, but we keep missing the SAR-11 big chunks. So we got to try again. Fission is what it is.
If Brian can find a large chunk of SAR-11's DNA, he will try to hook a whole microbe using the DNA fragment as bait. He will give the DNA fragment luminescence, like that of a firefly. With luck, the DNA probe will find a SAR-11 within the mix of marine microbes and enter it. The probe should then find a matching DNA sequence and, like a key in a lock, bind itself to this chemical sequence. The entire microbe will light up, and Brian will be able to see and photograph SAR-11 under a microscope.
The buffer?
I change it once every couple of months, yeah.
Now and then. Hey, Brian's got some pretty big DNA, it looks like.
Suddenly it seemed that Brian had found his large DNA fragment.
Well, it's looking good, but we have to look at the markers and find out how big it is.
The largest is 2200 kb.
But this one proved a blind alley. They were unable to confirm that it belonged to SAR-11. As so often in scientific work, they had to try again, and then again.
Yeah, just keep doing
end, it was Stephanie, using a variation of Brian's technique, who struck gold. 7 years after they first discovered a single SAR11 gene, the mystery microbe revealed itself and posed for its photograph.
Under the best microscopes, SAR11 is nothing more than a pinpoint of light. It's like looking up at night and seeing a star in the sky. And my feeling was like being the first person to land on the moon. It was like discovering for the first time organisms that you knew were tremendously important out there, that were present in numbers that went beyond the billions, and yet had not been detected at all by anybody previously on the planet. They're a lot smaller than most of the other bacteria out there. In many cases, they're 1,000 times smaller. You could put 1,000,000 billion of them into a tablespoon. What's it doing out there?
What is SAR11 doing out there? That's a question that applies to the vast majority of microbes on the planet. We know they have great power throughout the globe, but we often know few of the details. When we do, the details can be amazing. For example, microbes in the ocean affect the temperature of the planet. Ocean algae produce a sulfur compound which escapes from the ocean into the atmosphere. In the atmosphere, the sulfur particles cause moisture in the air to form cloud droplets. With many droplets, it's a very white cloud. With just a few, it's dark. The degree of whiteness affects how much of the sun's light and heat are reflected from Earth back into space, one of the things that controls the Earth's temperature. So, microbes have the power to affect climates. And it's always been that way. Long before there were animals or plants or fish or insects, when dinosaurs were still hundreds of millions of years in the future, microbes transformed the Earth's atmosphere from an unbreathable mixture of noxious gases to the oxygen-based air used by all the larger life forms that were to follow. Microbes have the power to control the evolution of life on the planet, but our understanding is only on the first page of the Book of Knowledge. And this was dramatically proven by Biosphere 2. What did go wrong there? We know that the oxygen levels dropped by almost a third in only a few months, causing the extinction of many species within Biosphere 2. Yet a conscious effort had been made to introduce plant, animal, and insect species that would successfully form a food web, a web of life. But microbial species were added almost inadvertently, most of them, like Earth's, virtually unknown. The microbes did what they're supposed to do. We created an imbalance by overfeeding them. Other factors probably came into play, but there is a strong suspicion that insignificant part, one of humankind's most ambitious experiments, was brought down in effect by a compost heap. Over-fertilization of the soil created a microbial feeding frenzy. reproduction increased dramatically to billions of trillions. This impacted the oxygen cycle. One microbe hardly makes a difference, but trillions of rapidly reproducing bacteria consumed a huge amount of the oxygen within the enclosed ecosystem. Was Biosphere 2 a failure? Yes and no.
Biosphere 2 gave us an opportunity to begin developing ideas that we need to understand how our own biosphere works in detail. Can we afford not to understand what microorganisms are and what they do? Up till now, we've gotten by without understanding them. But we take for granted that Earth's biosphere will continue to hum along into the future without the sorts of significant problems that cause almost devastating trouble for Biosphere 2. Can we permit them to remain a mystery? Before we begin predicting how our own biosphere will respond to human influences, we have to find out what these organisms do. Without the microorganisms, there could be no plant life, there could be no animal life. We would disappear.
The message is unmistakable. The microbial world is far more powerful than we sometimes realize. We've seen this power before. When the atmosphere of the whole planet was transformed with oxygen, it was microbes that did it. When a forest ecosystem regenerates after raging fires destroy it, it's microbes that spin the new web. It is microbes that enable every morsel we eat, every breath we take. There are always lessons to be learned. After all, wisdom begins at the moment we accept what we don't know, especially about the true keepers of the biosphere. Next time on Intimate Strangers, follow the story of a virus outbreak, step by step.
We knew we had a problem with some kind of disease that looked like an infectious disease. It had a very high mortality rate.
At the same time, discover why our bodies need microbes to stay healthy.